Great. Good morning. Good morning. Good morning. How are we doing? Surviving the gray? So today for class we are going to first do a lecture portion and then some small group activities, and then for the last section of class, like the last half hour, we're actually going to break into groups based on kind of our project interest areas. And we're going to get together and we're going to have a time to talk about how our projects are going and share ideas and kind of for people to get together a little bit around that. So that's our order of operations for today. Does that all sound good?
Yes, ma'am.
Awesome. So I am not going to waste any time. I am going to turn us right over to Matt Jones. Matt is the horticulture agent for Chatham County. He's like me, but in Chatham. He is an excellent teacher. If you ever see a class that he's offering and you are even vaguely interested in it, go. I know it's a drive. It's fine. It's worth it. It's a beautiful drive. So yeah, just an excellent teacher, excellent resource, always someone you can reach out to, and just a pleasure to work with too. So with Matt, take it away.
In reality, what he's not saying is that I'm rather disordered and diseased, so that's why I'm giving the talk today. But my plan within my section today is we'll do a crash course, a lecture kind of in diseases, diagnostic methods, and some on research methods. Then after the break, we'll come back and do some hands-on exercises and testing, mainly those diagnostic skills, both group research activities and then a full-on simulation where some of you will get to play the client and some will get to play. Oh yes. Hello all. Hello camera. Where there'll be a full-on simulation with some of you acting as clients, some of you acting as will be master gardeners. So a disease in this case is any a problem that is caused by some pathogen or other biological organism, and a disorder is more commonly referred to a problem caused on plants by some abiotic or non-biological issue like the environment, growing conditions, planting methods, etc. But to first know what is disordered or disease, you have to understand what is healthy, and what looks to be healthy on plants can vary among different plant species. So for example, on the left there is this small brown growth at the tip of a juniper. Don't worry, that's not a fungus, that is the male cones. So those are the structures that release pollen, so those are perfectly normal. Or something like this yellow honey locust here. All the leaves are yellow all the time. That's a condition of that cultivar. It's supposed to look like that. And same thing with variegation. There can be plants that have discolorations on the margins of the leaves or between veins, also quite normal. So know first what normal is. There are also things that can look strange to the untrained eye, but are not diseases or problems at all. So, for example, there are organisms called lichen, which are partners between fungi and algae, that are epiphytic, so they grow on plants, but they don't infect or cause any problems to the plants. In some cases, really a heavy accumulation of lichens can be an indication of slow growth, which could be a sign to explore further something else may be happening in the landscape, but for the most part, lichen themselves do not cause any problems. Or if you look on the underside of fern leaves, you might see strange, freaky-looking growths, right? Oh, I have a laser, I forgot. But those are perfectly normal too. Certain times of year, ferns will produce sporangia. So these are sori, which are groups of spore-producing structures that are perfectly normal in how ferns disperse propagules in the environment. And other things, like certain trees look weird when they exfoliate bark. All agents have had at least once in their life, what is wrong with my birch tree? Well, that's perfectly normal. It just looks different. So know what normal is. Also, there are some things that can look scary, but are also not only not harmful, but also normal. So there are sometimes in mulch beds, or beds that have mulch in them rather, you'll see suddenly this appearance of a scary-looking organism. This is called the dog vomit fungus. So you can look it up in extension literature, dog vomit fungus. It's not a fungus, it's a slime mold. But this is a slime mold that for much of its life is exploring, they are exploring and breaking down organic matter. And then under certain conditions, they will coalesce and produce this structure, which disperses the spore-like structures to allow them to spread in other places. So this is perfectly harmless. It won't harm anything at all. In fact, it's doing what mulch is supposed to do, which is decompose and add more organic matter to the soil. So there are many things that are not problems. Don't assume everything that looks unusual is necessarily a problem. If they are problems, though, we do, as I said, divide problems into two categories. Abiotic disorders, sometimes called abiotic diseases, and biotic diseases are sometimes just diseases. So abiotic disorders are those caused by some environmental stress or some cultural factor, which we'll explore in a second. Often, though, it is just simple user error. The problem is the people. And then biotic diseases, or true diseases, are caused by some infectious agent, some pathogen. And within plant pathology, this is primarily fungi and fungal-like organisms, secondarily bacteria, and in other cases viruses and even nematodes. Nematodes, yes, they're an animal, but due to the funny history of plant pathology, often nematode infections are part of the discipline of plant pathology. So there are the way you can look at a plant that has problems and try to deduce if it is an abiotic problem or a biotic problem based on patterns you see on the plant and patterns you see among other plants in the landscape. Generally, abiotic disorders tend to appear suddenly, whereas diseases tend to appear relatively slowly. It takes time for the infectious agents to spread in the tissue, for example. Abiotic disorders tend to affect many species at once. If there's a drought, there'd be multiple species that are not closely related that could be affected. However, diseases tend to be host-specific or relatively host-specific. So they're only able to overcome the defenses of a set group of plants, and often those set groups are closely related to each other. Abiotic disorders tend not to spread. Often it's an acute, sudden event that happens once and dissipates, and then the effects of that are seen in the landscape relatively quickly, whereas diseases, as I said, spread slowly. Abiotic disorders tend to have more geometric or linear patterns, so if there was a misapplication of herbicide on one side of the sidewalk, you're going to see a line of damaged turf or whatever. Whereas with diseases, because the infection event was probably pretty random, whatever dispersal agent, air, water splashing, etc., was relatively random, you might see hot spots, random areas that are having the disease. Then if it's spreading, it might spread from those random initial colonization points. And often with biotic diseases, not often, but not infrequently, you might see a transition from healthy to recently infected to less recently infected tissue. So a classic example would be a spot that is brown at the center with a yellow halo that's surrounded by green leaf tissue. That's a strong indication that there is a pathogen spreading in that tissue. Effectively, and maybe not a bad heuristic or some operational idea when you go into these sorts of things, diagnostic processes, is to start under the assumption and let's rule out abiotic causes first. Let's try to just get all the information you can from the client, understand the situation in the landscape before you automatically assume it's a disease. Most people want a disease as an answer, because then they think if there's a simple disease answer, then there's simply something I can spray to make it magically go away. The same erroneous attitude we have in medicine too. Everything is, some magical problem that has one easy solution. So maybe start with the assumption, get as much information you can to rule out abiotic disorders first. Examples of abiotic disorders are often, as I mentioned, user errors. This could be poor choice of plants in a certain environment or habitat, so poor plant selection. A plant that thrives in full sun, that's planted in deep shade, is going to be more likely to have problems and may, in fact, down the road, be more susceptible to diseases because that's a stress situation. It's not uncommon to see improper planting depth on some species that are prone to being really sensitive to that. For example, green giant arborvitae is really grumpy if you plant it any deeper than than the container or the bulb and burlap root mass. In other cases, if there's a problem with, say, a flowering shrub not blooming, well, if the buds form on previous year's growth and it flowers in the spring, if they cut back all the growth from the previous season in that winter, suddenly, yeah, you're not going to have any flowers because you cut them all off. So understanding bloom time and that sort of thing can also be a cause of some observed problems, user errors. Then others are more environmental, so too much heat, too much cold, or the temperatures are too high or too low. That can come in the form of actual freeze damage, so water freezes, ice crystals, rupture cells, etc. Other plants are simply sensitive to colder temperatures that are still above freezing. So a lot of warm season vegetable crops, for example, are native to more tropical environments and get really grumpy or grow very slowly, get stunted if it's even as cold as 50 degrees or lower, not necessarily 32. In other cases, there can be like too much sun or too much heat that will cause tissue desiccation, or a common issue with peppers is fruit sunburn. So if a pest comes along and chews off some leaves, suddenly the fruit that was formerly partially shaded by those leaves is exposed to much more light, and that can damage the tissue before the tissue has a chance to develop the right pigments to absorb all that light. Too much or too little water can also be a major cause of, a frequent cause of issues. Increasingly in our area, this can take the form of droughts in the fall, especially after planting. Frequently our driest months now are October and November. Sometimes our hottest months are October. And so that's a common time to plant, the idea being that you allow the plant to grow roots during the winter, because it's still not completely dormant, so that there's a greater root mass the following summer. But they can be sensitive to drought if you're not remembering to water them. Or say a plant that normally gets along fine, it's been a landscape for years, but there's been a long drought in October, November before it's completely dormant, and you will need to water, provide supplemental water. In other cases, too much water is bad. Plants can die from too much water in at least two ways. One, they need oxygen. Plants use oxygen just like we do. If they're growing and they're photosynthesizing, they're releasing more oxygen than they're consuming, but all their tissues consume oxygen for the same reasons we do. And so unless they have special adaptations, if the roots are constantly inundated with water, the roots won't have enough access to oxygen and will slowly die. Further, wet conditions will favor the colonization of those tissues by infectious agents like bacteria and fungi. There can be other things that manifest in that are not necessarily infectious agents or anything, but you might find that the symptoms are not necessarily, where you see the symptoms are not necessarily the indicator of where the problem is. So like with nutrient deficiencies, they can manifest in the leaves, but ultimately this is an issue with the soil and nutrient management. And sometimes if we misapply, especially synthetic herbicides, which are just different forms of salt, that can accumulate in plant tissues and cause injury too, depending on the species. I haven't seen much of that here though. I don't think I've ever encountered that in North Carolina yet. And even misapplication of herbicides or herbicide drift can be a major issue in some areas. I worked in Harnett County for a time, and probably 4 or 5 times in my 2.5 years there were homeowners who were next to a field or a farm, and the farmer sprayed an herbicide in a time he or she shouldn't have, and those herbicides drifted over into their vegetable garden and caused unusual symptoms. If that ever happens, by the way, you are not the arbiter of that. If that's ever an issue, they need to call the Department of Agriculture. They are the regulatory agency, and they have pesticide regulators that will handle that. So just don't get the lawyers involved there. Other issues can be mechanical damage, so a mechanical injury from using equipment or improper pruning, that sort of thing. Often this can manifest as line trimmer damage. So someone gets a little bit confident in taking some plants out close to the tree trunk and oopsie, I just ripped off half the bark. And that's problematic potentially for plants because on the outer edge of the plant tissue and stems is phloem, the cells that transport or translocate food made in photosynthesis. And so if you remove those tissues, there's no way for them to regenerate it on that injured side. So one way to get around this to prevent overconfidence is to apply a mulch layer and just not. Don't do that, please. Other random acts of nature can be a thing too. This is probably more common in a larger farm context, but lightning strikes and hail can cause kind of strange symptoms. It's not uncommon to have a random dead spot in a soybean field or something. It looks awfully disease-like, but when you look at the tissue more closely, it's just been absolutely fried by a random lightning strike. So what do diseases look like? Let's look at actual pathogens and how those symptoms differ, or how they appear in plant tissues, where they appear, and how they differ in from abiotic disorders. So a disease is often named partially by where it's occurring on the plant. Just like we have diseases of different organs, plants can have the same thing. There can be a disease of the fruit, of flower, of leaves, of roots, of stems, etc. So look at all potential parts, or even roots too. Look at all potential parts of the plant as the source of the issue. Within diseases, we tend, within the signs and symptoms that, I've ruined this here, signs and symptoms that give us the suggestion that there is a disease, we make a distinction between signs and symptoms. A sign is some direct evidence of the pathogen or pest. So in the case of insects, that would be you literally seeing a leaf-footed bug feeding on a peach fruit or something. That's obviously a direct sign. In the case of diseases, this would be more something like you clearly see evidence of the infectious agent. So in the case of fungi, often that is the vegetative bodies called mycelia, the little strands of cells that are growing on or in tissue. It may take the form of the fruiting bodies, so the spore-producing bodies of the fungi. And you might even be able, in some cases it's required in fact, to look at the spores under the microscope to help you identify the pathogen if you're a diagnostic lab. In the case of bacteria, we'll see later, they are much smaller than fungi, and they typically infect inside tissues, and they need relatively moist environments to live and divide, and so often you can see them oozing out if you sever the stem in water. So all those are signs, direct evidence that you see that an organism growing on the plant. A symptom is more likely though, they're easier to spot. They may be slower to manifest, but they can be easier to spot in the landscape as you see something is wrong with the plant. And it's, in fact, a symptom is some physical attributes that are a consequence of the pathogen infecting the plant. So this is really effects you're seeing on the plant is a symptom. And plant pathologists, in kind of a general sense, have different terms for different kinds of symptoms based on where they occur, but more so on their shape and form and what they do to the plant. So something like a spot, as you might imagine, looks like a spot. It's round, or it may not be perfectly round. It may be irregular, but it's a multiple spots on usually leaf tissue. Stunting just means it's growing slowly for some reason. Often this can be a stress in the root zone or maybe time of flight, etc. Rots refer to general decay, kind of massive, just brutal decay, not recoverable decay. So often we have rots on fruits, for example. Cankers refer to usually kind of elliptic lesions, often on stems, and they're often partially sunken, but not always. That's a term that gets thrown around a lot, usually inaccurately, but the example in the photo is a good example of a true canker. You can see how this lesion is a little bit sunken and rough and distinct from the healthy tissue. A wilt is not just meaning that the plant doesn't have enough water. It means there is a wilt pathogen is a pathogen that's growing in the tissue that transports water or transports starches. And because of that, it's interfering in the ability for the plant to take in water. So there may be plenty of moisture in the environment, but because the plant can't get water or other translocation, things that are translocated up the stem, it's wilting. And then if you cut open the stem, you can actually observe those infected tissues. So here, this the secondary xylem here is kind of brown. That's a good indication that you have a wilt disease, and those are not recoverable. That's deadly. Blights refer to they're sort of like rots, but on the whole plants, and usually refers to vegetative parts. So a widespread dieback of stems and leaves, usually asymmetrical. And then dieback is kind of similar to a blight, just more specific. So, dieback is a more general term to describe the fact that tissues seem to be dying from the tip inward. And then viruses tend to impart a mosaic or model pattern where there's a lot of like small areas, many small areas within the leaf that look different than normal tissue. So lots of little yellow spots where the viral particles have infected or ruptured the cells. But stink bug leaf feeding can sometimes look like that vaguely. Within plant pathology, we have a concept called the disease triangle, or if you want to get 3D, the disease pyramid. But the triangle is to imply that for a triangle you need at least two sides that are, you need all three sides for things to be stable. So if you remove any one of these factors, then disease will not occur. And those factors would be, you have to have a host that is susceptible to a specific pathogen. That pathogen has to be present, and the conditions must be right for that pathogen to infect the tissue. If you don't have all three of those, you may not have a successful infection. You might not have disease. So in some cases, we try to manage diseases by targeting one or more of these points in the triangle. Can you find a cultivar that is resistant to the disease, a host that is not susceptible, in other words? Can you do something to kill the pathogen or prevent the pathogen from spreading? Can you apply a fungicide that will prevent further infection? Or can you alter the landscape in a way that you are, say, making sure there's good airflow to dry out the leaves, that reduce moisture on the leaves, that in turn reduce the favorability of the environment to allow for successful infection. We covered this briefly earlier, but just to emphasize a few points, biotic diseases tend to be restricted to certain hosts. So if your rose has diseases, which is the rose, of course it has a disease, it's a disease pretending to be a plant, kind of like, well, most members of that family are like that. It's not going to spread necessarily to your gardenia or your lawn or to your dog. Okay, there's these pathogens only evolve to infect and overcome the defense mechanisms of a specific group of plants. You'll see a transition from healthy to infected tissue. So this is a bean plant with a fungal leaf spot. And so what happened is that a fungal spore landed on, I have this, sorry. Wow. A fungal spore landed on the leaf. It germinated, the mycelia started to grow, infecting and destroying tissue. So it spread outward in a roughly circular pattern. And so the areas that are the most brown are where the infection started, and then where it's actively infecting new tissue is this yellow halo, and then it hasn't gotten to the green tissue yet, but is likely to keep on spreading. Not all fungal diseases and most bacterial diseases don't have a halo, but if you do see a halo, that's a very good indication it's a fungal leaf spot. And then sometimes well, not sometimes, often, you can have infectious propagules, spores often, or in other cases, parts of the organism that are vegetative, but in hard bodies, that can remain in plant tissue or even remain in the soil. So for example, a sanitation is a good practice in some cases, especially in annual vegetables, that sort of thing, which are more susceptible to a lot of diseases. It's important to remove infected material, because if you leave the tissue, the infected tissue in the garden, it's more likely that that disease will appear the following season or infect other individuals at that season. And in some cases, those can be quite resilient. So this is a disease called southern blight, and it produces not only spores, it can produce spores, but often it reproduces by just making hard individual vegetative bodies from the mycelia. And these vegetative bodies, called sclerotia, are extremely durable. They can remain in the soil for a very long time. They're very hard to destroy. They don't decay easily. So often, if you have a disease like this in the soil, you're going to have it forever, unless you completely remove the soil. Bacterial root diseases are also like this. It's hard to remove bacteria. So if you have a bacterial wilt disease, for example, it's going to be in your soil pretty much forever, and you have to find another strategy. If you're into plant pathology, one could go into a lot more about how each of these types of infectious agents work. We'll just cover this briefly. So we tend to group them mainly, we're going to exclude nematodes in this case, but are they fungi? And then there are other fungal-like organisms. They're fungal-like because they grow sort of like fungi do, but they're actually more closely related to animals and some types of algae, but we won't, So I didn't add this here because there are a lot of examples in plant pathology where you're dealing with a water mold. And sometimes strategies that you would use to prevent a fungal infection may not be as effective on a water mold and vice versa. But in general, a typical fungus is not just a mushroom. A mushroom is a large fruiting body of certain types of fungi. Most of the body of a fungus is lots of strands of cells growing in the soil, growing in other organisms, etc. It's only in certain conditions will they produce the fruiting bodies that release more spores. So the spores can land on tissue. They're able to infect healthy tissue if it's a pathogen, whereas something like a bacterium, which is much, much smaller than a fungal cell, requires some sort of opening to enter and infect the plant. That can be a injury, mechanical injury, even something as subtle as when you plant a transplant into the soil, you could slightly break or scratch or damage a root, and if that bacteria is in the soil, it can infect that way. In other cases, they can enter through openings, and on the plant case of leaves, all leaves have openings called stomata that allow carbon dioxide in and allow water out for transpiration. So they can enter that way, so you can actually see the bacteria inside the stomata, that's quite a cool image. And then viruses require some sort of vector, so some animal to move them around. Viruses are just particles of DNA or RNA. They are biological but not alive. They're not metabolizing. But a insect, for example, serves as a good vector of most viruses and almost all viruses in plants. So they'll have it on their mouth parts, and they're just doing their thing. And when they're sucking on that plant, they may get the virus into the plant cells, and then the virus will then replicate within plant cells and rupture the cells and keep on spreading. So viruses need vectors. And so often management relates to controlling the disease vector or getting a resistant cultivar. Oh, I was going to say all this. So I said all this, now you have it in writing here. So fungi have hyphae, they grow in things, produce fruiting bodies when conditions are right, release spores, repeat the process. Most fungi are perfectly fine. They're critical to, in fact, life on Earth and carbon cycling, nutrient cycling, all of that. Others, and often they help with decaying that matter, and if they are only growing on dead material, they're called saprophytes. But if they are able to infect healthy tissue, they are parasitic in that sense. And so often, unless we're looking at a plant that has been stressed for a long time and is on the decline anyway for other reasons, if there's a disease problem on a plant, it's probably a parasitic fungus of some sort. Yes, they can do cool stuff, penetrate live tissue. They can cause any and all of the types of symptoms I mentioned earlier: diebacks, cankers, leaf spots, etc. And then there are others called rusts and smuts or scabs. But rusts and smuts especially do refer to a specific subset of fungal pathogens. So I would be careful in describing something as rusty unless in your research you've come across that one of the potential pathogens of that crop or species is a rust fungus. So that has specific meaning if you see it. Oh yeah, and this example, sorry, is a famous one. I don't have it today, but you will come across this, I'm sure, in our exercises. But there's a rust called cedar apple rust, and as the name implies, it actually has two hosts. So in one stage of its life cycle, on the leaves of apples or others in that family, malloid roses, you'll see leaf spots, and they produce a certain kind of asexual spore. And those spores can only infect not other apples, but they infect junipers. And when they grow in junipers, they produce a different kind of growth pattern. It looks pretty cool. It's like this big, spongy, tentacle little thing. Quite harmless to the juniper, by the way. This is not a major pathogen. This will never kill a juniper. But looks very different, but this structure then releases spores that will then infect other apples. So both of those hosts are required for that organism to complete its life cycle. But notice how different the appearance is and how different the effects are. This is pretty harmless. In apple orchards, this can be bad for reducing yields and in some cases even killing the tree. Bacteria, again, much smaller. Are much smaller, orders of magnitude smaller than fungal cells, animal cells, plant cells. They're a much simpler internal organization, and they do require moist environments to thrive and reproduce. They do require a wound or an opening to enter. Probably one you may encounter. Oh, please remember this image later. Hint, 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 hint. But here, there was wilting seen in a certain plant, and they cut open the stem. It looked problematic, but it was hard to determine if it is a fungal infection or a bacterial infection. Well, if you stick the cut stem in water in a clear jar and you see ooze coming out, hint, hint, that's definitely a bacteria. So that's called the ooze test.
Is that immediate? Like if you put it in the water, you'll see it immediately, or?
Oh yeah, you'll see it immediately.
Okay.
Yeah. And then bacteria can cause all sorts of issues on all parts of the plant. So they're not limited to just leaves or something. There are bacterial diseases of all parts of certain types of plants, is the takeaway here. Viruses, again, they have vectors, so they have like an insect. This is a whitefly that is feeding on something, I don't know what this is. Anyway, feeding on a plant, and in the process of consuming the phloem, spreads the virus inadvertently, and that multiplies within the cell and destroys the cells and then spreads to other cells and causes problems. These aren't treated. You can't give them a mRNA vaccine. And then often they're not really fatal either. If you're producing a fruit crop, they can be problematic because they can reduce yields pretty dramatically. But in an ornamental species, either live with it or move on and plant something else. In most cases, when this is a problem, you want to look for a hybrid cultivar that has been bred to be resistant to viruses. And many hybrid cultivars of vegetables are in fact resistant to a lot of viruses. So you'll see most often is just spottiness on the leaves, little yellow dots everywhere where the cells are dead. Especially though if it infects younger tissue, not fully developed tissue, that will cause distortions in how the leaf is growing and how it emerges. So that's also common. There can be spots even on fruit tissues in some cases. So this looks deceptively, I don't know, bacterial, but this is a viral disease of pepper. And in some cases, they even cause conditions called witch's brooms. And this is really caused more common by not a true virus, but a phytoplasma or something, but the same idea is there, that the growth releases, the presence of it releases a hormone that causes distortion and growth around growing points in the stem. All right, next, let's do the diagnostic process in a nutshell, and then we'll cover, I'm making great on time, okay. Then we'll cover some research methods, and then we'll have delicious snacks provided by the Extension Master Chair Volunteers Association of Durham County, which is why any of us are actually in Extension.
Yeah.
Okay, so I'm going to show you a diagnostic process, a kind of a 5-step process. This is not the only valid process, but this is the process that is in the Extension Gardener Handbook. So just to give you, if you need help, like knowing where to even begin, this may be a methodology to try out. So know what you're dealing with. Make sure you confirm that what the client is telling you is actually what the plant is. So if they are saying it's a cedar, well, maybe it's a juniper, because cedars, some junipers are called cedars inadvertently. If they say my shrub is diseased, that's not helpful. We need to find out what species of shrub it is. And so don't assume they know anything, have any idea what they're talking about either. Just be nice about it, That's okay. That's why they're calling you. Okay, it's for help. So don't be nice. And if you can, especially if it's a vegetable crop, find out what the variety is, the cultivar, the cultivated variety, because knowing that can help you rule out some diseases. If you have a Better Boy tomato, that should be resistant to 4 or 5 viral diseases, should be resistant to early blight, so you can just rule those out of immediately. Know what normal is. We talked about this already. So just understand what is normal for that species or that cultivar. I know, behold my beauty. Then look at the distribution of the problems or the symptoms both on the plant and in the landscape. So if someone just brings in a single leaf they've ripped off the plant and tell me, what's this? That's not helpful. You need to have context clues for where it occurred on the plant and how it's distributed in the landscape. So where it occurred on the plant is it's starting on new growth. So the tips of stems, the tops of plants. Is it starting on old growth, the bottom of stems, the bottom of plants? Is it occurring just on the leaves, or is it occurring on the leaves and the stems? Is there a problem with the roots? What are the growing conditions? Is it occurring just on a single plant, or are there 3 or 4 nearby that are showing the same symptoms? Are those other plants the same species, or are they different species? Figure all that out. You need lots of context to be able to do this correctly. And we've already gone over kind of how these patterns vary between abiotic and biotic disorders, linearity versus kind of hotspots, appearing suddenly versus appearing slowly. I should add too, appearing suddenly is all relative, because often the clients might say, oh, it just appeared out of nowhere or suddenly. Well, that could be, they don't want to admit it's been a problem for a long time. It's like people with Y chromosomes when they go to their own doctor, yeah, I thought that was just a new thing. No, or they just noticed it. I mean, so in their mind, it just appeared, but it could have been on there for a while. So try to tease that out if you can when you're asking them questions. And then again, on the plants, the patterns on what organs are they occurring on within that organ. What are the patterns like? Are there symptoms on the margins of the leaves? Are they happening on random spots? Are they happening on one side of the leaf? Are they happening at the base of the leaf, on the veins, between the veins? Is it happening on one side of the plant? That's also an interesting clue. If it's all on happening on one side, usually that would indicate a biotic problem. That's just one infection point. How uniform is it? Like, is the whole plant showing disease symptoms that might be more indicative of a wilt disease or a root problem? But if it's just happening on one side, that can be maybe more likely a leaf or stem issue. Look for certain patterns, because sometimes the appearance of the spots, the appearance of the canker, the color of the infection point can all be clues to what it could be. And when you're looking for problems for specific plants, once you know what it is, often the names of the diseases that are known have the name of the organ in it. So tomato leaf spot, early blight of tomato, or root rot of gardenia, things like that. So use the name of the organ and as best you can, the description of the symptoms along with the name of the plant when you're doing searches for potential problems or explanations. Then in some way, your choice, but systematically go through the potential causal factors and start ruling things out. So again, we group these things into abiotic or biotic. If it's abiotic, is it a cultural problem? So the wrong plant, wrong place, planted the wrong way for the wrong reasons, or has there been some environmental stress, heat, sunlight, cold, heat, I said that right, etc. And then if it's not abiotic, is it a biotic issue, but is that caused by an actual pathogen, or is this an effect of some sort of insect feeding? And if that's the case, is that actually a problem or not? So have you had entomology yet? Okay, so you just a review on pests. This can be the signs of growth distortions. You can see slime trails from say slugs. You can see evidence of webbing or frass, the excrement of insects, or even shed molted skins or exoskeletons from insects. And then they can damage things more directly by chewing on them in many different ways, inside, outside, different organs that you talked about. And insects can serve as vectors, as I mentioned, and even something like fire blight is completely innocent. It's just the poor bees. They're just trying to get to get the nectar on that stupid calorie pair, and then they get the bacteria on their legs or something, and they go to the next flower, and then the bacteria get into the nectary of the flower, and that spreads into the stem and starts etc. So look for all those things. Rule those out first is one strategy before you start assuming it's an actual disease. But there are other approaches. You have to go in that step. Virginia Tech has one where think about its context clues first, and really, but basically this is going through all the abiotic issues first, like systematically, and then looking for signs and symptoms, and then sorting those by either being a pest or a pathogen, and then what kind of pathogen. The Florida system, they start, it's Florida, so lots of pests in Florida, many in Tallahassee especially, and so they, let's say, rule out the insects first, just make sure it's not some insect issue, and then consider abiotic issues, and then only after you rule those things out, go through the potential morass that is diagnosing actual disease. Okay, other just kind of recommendations when you're interacting with clients. Beware of any sort of leading questions like how much lime should I apply? You can only know that if you've done a soil test. So ask that first. And you need to how they know how to apply lime if they don't even know the soil pH or other soil parameters. Take all the information, be nice about it, but be able to understand that perceptions can vary and that memories are also faulty, can be. And then understand that you are here to answer questions from gardeners. If it's from a farmer who gets onto the plant clinic line in some way that needs to go to another agent in the county. And then also try to extract as much information as you can from them, because vague questions and vague information you can only give them vague and unhelpful answers. So extract as much information as you can from the client. And you guys may have this, if not I can share them, but most plant clinics or Ask a Master Gardener hotlines will have diagnostic sheets that will help guide you through those questions, or allow you to take those notes or remember all the questions to ask. You know, agents can't do visits to homeowners. There are only 90,000 houses in Chatham, many, many more in Durham, so no, Dashley cannot visit everyone's house. Okay, that's not doable. We can't endorse specific companies, but you can provide lists of recommendations or sources of stores and that sort of thing. And you can't tell them things without some information. It's like, I have a bug on my plant, what is it? Well, I need a picture, or I need a sample. I can't read your mind, etc. And pesticide stuff, you don't talk about that. You tell them to read the label. That explains how to use it, what to use it on, etc. And for insects, you know, that makes sure you're asking things, what they're occurring on, what the conditions are, and what it's doing. Is it really feeding on the leaves? Is it just nesting? That's usually not an issue in most cases. If you're identifying weeds, it's helpful to know the stage of maturation the weed is, because that can affect your management options, etc. And then you do need good pictures. This is an actual picture that I received, and you know, that's not very useful. So make sure they're good, high resolution. Make sure you get pictures that are in context. Again, you need to see how they're distributed on the plant. If you don't know that, that can really make the diagnostic process a lot more challenging. So close-up images, mid-range images, distant images, contextual images are all good. And a scale can be helpful sometimes. So just some other object in the photo that shows you the size of the lesion or whatever. And then there is a whole diagnostics chapter of the handbook that summarizes some of the most common problems. That can be useful sometimes, just to give you a big picture overview, but the number of potential problems in most species can be nuanced, so don't assume that if it's not in this chapter, it doesn't exist. It's a kind of a baseline. Okay, and then if you have you may have watched this already, but there's a kind of funny video that Lucy Bradley and a few others made like 15 years ago, that just is a way to train master gardeners not to panic. They get a phone call, and it's the most intimidating part of being a master gardener is answering those questions. So don't panic. This will give you some tips to do that. The link is embedded in the PDF of this presentation. All right, finally, yes, I'm doing awesome, I think, maybe. We'll go through some research methods. Wait, first, I'll stop. Be quiet, Matt. Any questions so far? Yes.
Briefly, the difference, how do we identify the different galls caused by pests versus galls caused by diseases?
Well, in part would be the location on the plant. So usually a disease gall is at the base of the crown, so meaning the base, the transition area between the stem and the roots. The most common is agrobacterium on some plants, and that's a tumor that's caused by a bacterium. So I don't know of any insect gall that occurs at the base of the stem, for example. Insect galls will tend to be on leaves or tips of stems. Also, because insect galls are for encouraging insect development, you can cut them open and find some evidence of an insect on the inside. I would say, if I was a betting person, if you see a gall, it's probably insect, that's probably harmless. Oh, certainly harmless. So don't be too concerned with galls. Crown and stem galls caused by bacterium usually are limited to not many plants that we have in our landscape. Grapes get them a lot. Some trees get them, but they're really obvious. So generally, if you see a gall, it's not a problem. But when in doubt, cut it open. What else? Yeah.
You and the book both mentioned certain bacterial infections in the soil are irreparable?
Uh-huh.
So what do you do?
You plant something else.
Okay.
So in practical, so that's the easy answer. But
right.
in a practical sense, you need to have a resistant cultivar.
Okay.
And in the case of something like a tomato with bacterial wilt, that's only achievable through a grafted plant, we don't yet have a resistant cultivar that is both resistant and is worth eating.
Okay.
So you have to actually fuse a resistant rootstock that can tolerate the bacteria with a tomato that is worth eating. And you're in the great spot for that because there's lots of good history and practice of that in Durham County. But yeah, that's kind of the main thing. But if and otherwise, go ahead. Sorry.
And your sale?
Huh?
Your sale?
Oh yeah, and we sell them too. So stand by for that. That's our only fundraiser is grafted tomatoes. So if you want to know about that, please go to the Chatham Extension website. And on the bottom right, like with any county website, there's a place to add your name to a newsletter. You can be notified of that. But I'll share it with Ashley too. So yeah, anyway, so yeah, when those bacteria, go ahead, sorry. You
Were
need
you
to.
getting at what to do with the soil?
Yeah, essentially, like, so bacteria are also relatively host specific, so you're saying.
Yeah, so you
you're
could
not
plant
really
other things.
remediating the soil in any way.
No, there's no way to remediate the soil at that point. You can, I mean, there are ways you could potentially maybe try solarization,
Right.
but bacteria are just finicky things, and they can occur in stay. One cell could be within one little soil particle and be fine, and then voila, it's still there. So from a practical perspective, it is hard to eliminate root pathogen bacteria from soils. Yeah, there's no bactericide. I mean, bactericides are not really a thing in most crop production. And for fire blight management, you can apply streptomycin, but that's usually for farmers and not a thing you would do as a homeowner. But bacteria, if they're there, they're there.
This is a very basic question.
That's okay.
But going back to when you had the diebacks, they look like a camellia or something, you
Uh-huh.
got that one little piece of dieback.
Uh-huh.
And also with the leaf that has the spot. Are those things that could be solved by just cutting off whatever is dying back or the one leaf? Or is that something then that becomes systemic in the plant?
In many cases, yes, it can be. You can reduce the degree of infection by removing the infectious part. However, sometimes the extent of infection is more than meets the eye. So often with a dieback situation, you end up cutting several inches into healthy tissue just to make sure, because the pathogen could be deeper than it appears. And in other cases, with some spots, then they could be quite extensive, so you'll end up taking off all the leaves sometimes. So, yeah, but yes, that's a perfectly valid. It's called sanitation method in many cases to remove the infected part. And if it's a bacterial wilt, though, again, because bacteria spread so easily, the example in this case would be fire blight. Every time you make a pruning cut, you have to sterilize your pruners because you likely have bacteria on the pruners, and you're just making it worse if you don't clean your pruners. Fungi, that's not always as much of an issue, but it's not a bad idea to just practice good sanitation, not only on the plant, but on the tools and yourself when you're interacting with plants.
And how do we sanitize our tools? I mean,
You can use 10% bleach solution is fine. Just make sure you dry so it doesn't rust your tool. Lysol, ethanol, or isopropyl alcohol also work quite well.
okay.
Mm-hmm. Mm-hmm. Yeah.
You had said with blight and biotics in general, sometimes the symptoms are asymmetrical.
Uh-huh.
What drives that? Is that
Based on where the infection started. So it's kind of a random pattern. So the spores blew in a current of air, and it hit one, and it could have literally been one leaf. And it started from there, spread down the stem, and started infecting that side of the plant. Now, if it's a root issue, that might still show up by the entire plant, because if the roots are infected. Although there are cases where sometimes some root infections will manifest in funny ways asymmetrically on the tree. So there's a very loose, R squared of 0.4 correlation between the side of a plant that's infected being close to the side of the roots that are infected, but that's not great, not that super reliable. Yes.
If you do pruning to remove the disease
Uh-huh.
of a plant, you know, if it's bacteria versus a virus versus insect, pest damage, or fungi, I guess, does that change, like, whether you would recommend the foliage be like bagged and put in the bin versus like it's okay to compost it, you know, in terms of the persistence of that?
Yeah, it sort of depends on how good of a composter you are. So the rule of thumb is typically if you can get the core of the pile where you put the stuff in eventually, gets up to I think 136 or 140 degrees, something like that, then that is guaranteed hot enough to kill, for all intents and purposes, any pathogen. If you don't think if you're just cold composting through slow decay, which is perfectly valid and fine, decomposition happens, so composting can be very easy. But if that's happening, the temperatures may not be high enough to necessarily kill those pathogens. And the persistence varies. I would, a fungal fruiting, like a fungal hypha or a fungal spore or a sclerotium is going to be more resilient than a bacterial cell in that case. But overall, I would just use caution, and if it's a major issue on that plant, just try to not put it in your compost pile. What else? Did that actually answer people's questions, or was I sounding like a politician? that this point, push me back if I didn't quite get there. All right. Do we need? So I would like to. So the last part of the lecture will just take about probably 10 minutes, 15 minutes. Can we power through that biologically? Okay. And then do you mind starting the break at 10:15?
Sure.
Ish. Okay, quick, okay. So finally, now, where do you even begin to, if that's not a problem, I can wait till 10:30.
No, it's good.
Okay.
They will be warned.
Okay. The mob is coming. So how do you find information that is valid from extension and relevant to answering the questions you encounter? And this applies not just to disease diagnosis, but any sort of information that a client may ask about. So our whole reputation is built on the fact that we're part of the universities, and we use science-based, research-based information. We're not going just whatever we can find on the vast end of civilization entity that is the internet. And when we're looking for research-based, science-based information, and extension, the way it should work is because in reality it's another matter, but in theory, on campus we have subject matter experts. We have professors that research these things, and they disseminate that information down to the agents, and we provide it to volunteers who help disseminate it to the public. And in theory, all this is peer-reviewed. It goes through the typical scientific process, research process, etc. As long as it's still funded, but we'll go. So ideally you want, it's just a question, valid question, no money, no research. Research-based information is the reason extension exists is to disseminate information from the universities to the public. And these are done through the land grant universities in each state. And every state has at least one, sometimes more than one. And if it's like on the west of the Mississippi, often if it has the word state in it, it's probably the land grant, but not always. But anyway, knowing what the land grant university is in each state can be helpful sometimes, so you can narrow down your searches further. But in North Carolina, that's NC State, NC A&T State, and then it's perfectly valid because we work in a similar region with similar problems to get stuff from Clemson, which is the extension university in South Carolina, and they have a lot of great publications. Rumor has it that 15, 20 years ago they laid off a bunch of staff because they couldn't afford them, and then put some of that money into developing a lot of these great fact sheets, so we can benefit from that sacrifice. But so if you're searching Clemson, you can search Clemson HGIC, and that's the Home Garden Information Center. That will bring up all their fact sheets. But Georgia, UGA, is the Extension University. In Virginia, it's Virginia Tech. So for really specific problems, that might be good to kind of stick to the southeast. But it's not necessarily wrong to, if you find a great publication that explains your symptoms, that's from University of Minnesota or Michigan State, that's fine. Just understand that in that publication, if it talks about we have one generation of the pest because it's only above 40 degrees for 12 minutes, you know, in northern Minnesota, then in the south, that's going to be, you could have many generations of that same insect because it's so much warmer for so much longer. California has a lot of great information, but very different system out there, right? They have, it's a Mediterranean climate, which means in the summer and it doesn't rain. Like, it doesn't rain for half the year. The raining season is the winter, and so that's one of the reasons it's so easy to grow stuff out there, is because when a lot of plants are growing, like peaches or whatever, there's very low disease pressure because it's so dry. So, but there's still some good information there. Just be wary of some, the pests can be quite different. We are in an area that is wet and warm. That means lots of organisms like to hang out here. So we have probably the highest disease pressure and insect pest pressure of anywhere in the country. But it doesn't have to necessarily just be from extension. A lot of the arboretums, and botanical gardens provide good information and often they're associated with some university. Oh, I forgot. I always forget that. I guess you guys have something here in Durham. But yes, that one is valid too, of course. And then, scientific societies, peer-reviewed journals, government agencies, for now, all have, in theory, research-based information. So what is not good information? My grandma always said, I can, you put some oatmeal and strychnine and motor oil on the tomato and it makes everything good. All that stuff is not going to be helpful, okay? You need to back up everything you're saying with some sort of research-based information. And don't trust some of these commercial websites. You never know what's on there. In fact, most of them are terrible, and maybe with some AI stuff, they're going to get even worse, because that's all that will happen with AI. And so, for some reason, I don't know if it's still the case, but certainly 5 years ago when I made this presentation, a lot of, there's something called SFGate, SanFranciscoGate.com, somehow had lots and lots of gardening articles, and I think they were mining extension publications and aggregating them, but you don't know where that necessarily came from. So, sometimes maybe this can be inspirational. Like, if you're really desperate and there's just not extension information on some really specific weird issue, but you get an idea that's inspired by something on here and that leads you to some other valid publication, then that's okay. But don't use this as a common way to find answers, okay? So look for extension stuff. This was the only CC licensed image of witchcraft that I could find, so it worked. Okay, so my top suggestions for doing this would be, historically, would have been going to extension.org/search. So this is just a customized Google search that just looks at extension websites. But the entity that oversees this website has declined a lot in terms of funding and its scope. Google has threatened a few times, apparently, that they're not going to do this anymore. And so this may not work forever. So instead, you might need to use more precise Google foo. And to do that, you would add usually .ext or .edu or extension or some abbreviation for the university. Any of those things are valid ways to make sure it filters through some of the nonsense. And notice too that Google now is using LLM AI stuff for their searches, which don't trust that. Okay, use the meat brain to delineate, or what I'm trying to say, make distinctions on what's good information and what isn't. So add any of these. With NC State, it's probably more helpful to add NCSU because that's what's in the URL. And that may also apply to other states too, like VT for Virginia Tech, or OK State, or Purdue, or whatever. Just knowing, adding that will improve your search quite a bit. Then you can look at the handbook. You know, we have some diagnostic tables in Appendix C, and I also love, anytime you're answering any question, make sure you mention the handbook or refer them to the handbook. Maybe embed URLs in the email so that the client learns to use this as a resource. All right, I think let's do a break. Oh, I'm really early. Oh, God. How about I explain in the meantime, so I don't scare everyone over there. We'll explain what the activities are. So you have before you, may I borrow your samples? So you've got the PowerPoint, of course, and then you have 2 sets of case studies. So we're going to divide into groups of 3 to 4-ish. And for cases that are numbered, cases 1 through 5, just work together using your new research skills to try to find and diagnose these issues. And so for each case, the front has some images, and the back has contextual information. And that should, for the most part, be enough to diagnose it if you find the right resource. If you think of something that is not on this side of the sheet, just pray, and then suddenly a godlike creature, being actually myself or another master gardener, will provide divine inspiration. So if we miss anything, then we'll help you out. But see, the idea was just work through these. You don't have to go in order. In fact, I might reorder these. Case 1 is a little tricky, but maybe if you want to work gently into this, start with case 2 or case 3, and then anyway. So do that as a group, and we'll spend maybe 15, 20 minutes, 30 minutes on that section. And then, time permitting, we'll move on to the diagnostic exercise. So this is for cases A and B. So case A is on the front, case B is on the back, or whatever. And you've got images, and you have some misformatted, descriptions of the question, like the client would send you in an email. Most of the time, it shouldn't be mocking, but sometimes, yeah, some of them might. Yeah. Anyway. So you have that. So in your group, some of you are going to play the role of a master gardener, and one of you will play the role of the client. And that person will have secret information, esoteric, occult information. And you have to be able to extract this kind of information out of them by asking the right questions. And so they, as the client, will have a cheat sheet so they can hopefully answer the questions you're asking them. And with those contextual clues, will allow you to diagnose what the problem is. Does that make sense? And if you want to, you can switch it up. Maybe for case A, one person can be the evil client, and then case B, someone else can. Because it is fun to torture the master gardeners. That is actually why I do this job. So that's the idea. Does that make sense? Okay. Sorry. I return everything I stole? Okay. So, release the mob. Feast. Oh, wait.
Sorry, I just have one
Yeah,
other
sorry.
question, just a clarification from something
Sure.
you said earlier. You're talking about the fungi biology.
Uh-huh.
And saprophytes and parasites. And
Yeah.
you said parasites can infect healthy plants?
Yes,
And saprophytes,
just decompose dead tissue.
okay, so they're feeding on something that's already dead. They're
Correct.
just decomposing. Parasites can infect something that
It's still alive.
I thought I understood, but I
Yeah,
just
sorry,
wanted
and I
to
kind of
sort
went through that.
of,
I went through that clumsily. So yes,
clarify.
yeah. Saprophytes only consume dead tissue, and then true pathogens, parasitic-like fungi, infect living tissue.
Great.
Other question?
So
Yes.
then, I'm just thinking mistletoe, so will that
Oh,
ultimately take
that's a
over
great
a
example.
tree and kill it?
So mistletoe is a hemiparasite. It grows on other plants, but it does still photosynthesize, so it does generate its own starches, its own food, but the root tissue steals water and nutrients from the tree. So a really heavy infestation of mistletoe can be stressful on the tree, but it's also, mistletoes are cool.
So it's symbiotic, well.
Well, it's not symbiotic. No,
symbiotic.
it is parasitic. It is parasitic. Yeah. So it is causing a decrease in the what's the evolutionary word I'm looking at here? The fitness of that tree.
So stresses.
It can stress the tree, but if you it's beautiful. It's part of life. Stress is life.
How about Spanish moss?
Oh, excellent question. So while mistletoe is a parasite, hemiparasite, because it's photosynthesized, but Spanish moss is an epiphyte. It's a plant that grows in other plants. So it's not infecting the plant at all. It's just using the plant as a surface to grow on. Most orchids are epiphytes in the tropics. What else is a good?
So why do you see Spanish moss on dead trees a lot? It seems to kind of overtake a dead tree.
It may be because if a living tree is growing, things are being shed. So bark comes off, branches break, but the tree still grows. And then if it's dead, the rate of shedding decreases, so or stops. So you get more and more Spanish moss growing, but it's just using it as a place to grow. Because if you're not having to devote energy into making rigid structures to grow tall and get sunlight or whatever, like vines and like the Spanish moss, you can divert more of that energy into growing faster. So they rely on the other plants to do the role of getting up high and reaching sunlight, etc. But mostly harmless. I mean, in all cases, it's just part of the environment. Yeah. And it's actually, Spanish moss. Is it a moss? No, it's a bromeliad. So it's closer to pineapple than it is to actual moss. So always use Latin names. Common names are lies.